Welcome to Cytek Bioscience's video tutorial on best practices for controls. In this video, we will outline the requirements for creating good controls in flow cytometry and how those apply to Cytek's full-spectrum profiling systems using Aurora, Northern Lights, and Aurora CS instruments. By the end of today's video, you'll be able to answer the following questions. What is a reference control? What are the four main requirements for preparing optimal reference controls? Why is reference control quality important? Why is it important to have an accurate, unstained reference control? To get started, let's review what a reference control is. A reference control is a sample stained with a single fluorochrome in order to define its emission pattern and spillover on the cytometer. In a multicolor assay, it is necessary to have a reference control for every fluorochrome that is present in the sample. In addition to the single-color reference controls, an unstained reference control is also necessary to measure the autofluorescence in the sample. Spillover occurs when a fluorochrome emits light into multiple detectors. In conventional cytometry, each fluorochrome is measured at its primary emission peak by a single dedicated detector. Any spillover by this fluorochrome into detectors dedicated to other fluorochromes would be a false positive signal. Here, we can see the emission of Phe and Pe when excited by a blue laser as they are measured in their dedicated detectors. We can observe Phe spillover into the Pe detector, as well as Pe spillover into Phe's detector. To accurately measure the amount of spillover between fluorochromes, we use single-color controls to generate a spillover matrix using a calculation called compensation. When using Cytek's full-spectrum profiling systems or any other spectral cytometer, rather than focusing on the peak emission, we measure each fluorochrome's full emission spectrum when excited by the onboard lasers using a series of detectors. Here, we can see the full spectrum signatures for Phe and Pe when acquired on a 3-laser Aurora system. Full spectrum profiling provides more data points and results in a more accurate spillover matrix, calculated using a mathematical algorithm called spectral unmixing. Another way to think of unmixing is the deconvolution of each individual fluorochrome signature from the total combined emission spectrum. The single-color references provide each fluor's unique spillover and contribution to the total signal. This includes fluorochromes, as well as the autofluorescence from the unstained cells or particle. For both conventional and spectral cytometry, the reliability and accuracy of the data depends on accurate measurement of spillover obtained through high-quality reference controls. Now that we understand what a reference control is and what it is used for, let's discuss the four main requirements for establishing high-quality reference controls. Keep in mind, these best practices apply to all flow cytometry experiments, whether they are spectral, conventional, low parameter, or high parameter. These requirements are: 1. The positive population must be as bright or brighter than the multicolor sample and should be well separated from the negative. 2. The positive and negative populations must have identical autofluorescence. 3. A sufficient number of positive and negative particles must be recorded. 4. The spectrum for each control must be identical to that in the multicolor sample. Beginning with requirement 1. Spillover can be estimated as the linear slope between two fluorochromes. This estimation is more accurate when there is greater distance between the negative and the positive populations that form this slope. When the fluorescence intensity of the negative and positive particles is measured and recorded, the system automatically computes a linear fit to these two data points and can accurately depict data from particles that fall within this range. If the positive particle in the reference control is dimmer than in the multicolor sample, the linear fit may be inaccurate, which can cause an error that is visually apparent as an unmixing error. This appears similar to a compensation error in conventional cytometry. Remember that the reference controls should be as bright or brighter than the multicolor sample. In this example, we show a CD57 BV605 antibody stained on both beads and cells compared to the multicolor sample. Even though beads are a convenient tool to create reference controls, they may not always meet this requirement, as seen here. The cell-based reference control, however, is as bright as the sample and will provide accurate unmixing. We can see this effect demonstrated in an IgM BV510 reference control. When the reference is gated on the dimmer BV510 positive cells, there are errors present in the brightest IgM positive populations of the unmixed data. When the reference is gated on the brightest IgM positive population, accurate unmixing is achieved. Next, we have requirement 2. The positive and negative particles in a reference control must have identical autofluorescence characteristics. The spillover of a fluorochrome is calculated from both the positive and negative populations. It's important to remember that the negative population's fluorescence is not 0. Not only do cells and beads have very different autofluorescence characteristics, as shown here, but different treatment conditions, tissue types, and even bead brands can all have unique profiles. In order to achieve accurate unmixing, it's important that the negative and positive populations have matching autofluorescence. For example, here we show a CD3 BV570 reference control stained on cells in which beads were added and used as the negative population. When unmixed, there is a clear unmixing error in the CD3 positive population. However, when a matching cell-negative population is used, proper unmixing is achieved. Notice that not only is the CD3 positive population resolved, but the negative population is also corrected. In some cases, a sample may not have a proper negative population to match the positive. For example, when staining whole blood with CD45, all lymphocytes are positive for this marker. Thus, there is no matching negative population. To work around this, Cytek's SpectroFlow software allows you to assign a separate unstained sample to be used in place of the internal negative population for a given reference control. As you can see here, in the software we have selected an unstained sample made from the same cells to serve as the negative control population. This feature can be used in a number of ways to offer flexibility when creating reference controls. Here are some examples of positive and negative particle combinations that should or should not be used together. Firstly, we can safely match a tube of unstained cells with positive cells, or match unstained beads with positive beads. These matching populations can also be run within the same tube. Conversely, we would not want to mix a cell reference with unstained beads for the negative particle type, or the reverse. Next, requirement 3. A sufficient number of positive and negative particles must be recorded. For each reference control, you need to record enough events to generate a statistically confident estimation of the spectral signature. In SpectroFlow, visual feedback from the spectral plot can help inform if enough events of each population have been recorded. A minimum number of 200 events in both the positive and negative populations is required to calculate unmixing. If you attempt to unmix with too few events in the gate, the SpectroFlow software will display an error message indicating which control and population did not have enough. Remember that we want to gate on the brightest cells, which can sometimes be a rare population, as shown here for CD56, so make sure to acquire enough events to satisfy this requirement. For the final requirement, we'll discuss how the spectral signatures in the reference controls must be identical to the spectrum in the multicolor sample. When the spectral signature of the reference control matches that in the multicolor sample, accurate unmixing is achieved. In this example, CD8-CFluor-B532 is properly unmixed because the reference control meets this requirement. What are some things that can cause the signature to differ between the reference control and the multicolor sample? One source could be the use of a very similar but not identical fluorochrome in the reference control. For example, you are running a multicolor assay that includes GFP and PE. However, the reference control for the GFP was instead made with FITC. Though these two fluorochromes have very high similarity, they are not identical. By viewing their normalized spectra, we can see small differences. When the GFP and PE multicolor sample is unmixed using FITSI as the reference control, unmixing errors are present. When a proper GFP control is used, accurate unmixing is achieved. There are other factors that can alter the signature of a fluorochrome. For example, an antibody labeled with a given fluorochrome can sometimes produce a different signature when bound to compensation beads compared to when it is bound to cells. Here, we compare the same BV711 antibody stained on cells and beads. We can observe differences between the two BV711 signatures in the B10 and R4 detector regions, which lead to unmixing errors in the data. For example, there is no difference between the two signatures in the V10 region, thus unmixing with BV650 is accurate. Meanwhile, minor differences in the B10 peak produces minor errors with the per-CP eFluor 710. Lastly, a greater difference at R4 causes a more significant error with APC R700. Because of these effects, bead and cell controls should be validated for every assay. Certain buffers and treatments can also alter a fluorochrome's signature, such as those used when processing stained samples with fixatives or permeabilizing detergents. To ensure your reference controls are accurate, treat them with the same buffers and reagents as your multicolor samples. In addition to reagent interactions, some fluorochromes may be sensitive to storage conditions. Shelf life, light exposure, and temperature can all drive changes in the spectral signature. To minimize variation, check that all reagents are consistent between your reference controls and multicolor samples. Special considerations must also be taken when working with tandem fluorochromes. Lot-to-lot variations of the donor-to-acceptor ratio of a tandem fluorochrome can result in slightly different spectral signatures. Therefore, when using a tandem dye, the same lot of antibody must be used in both the multicolor and reference control sample. Now that we've reviewed the requirements for single-color reference controls, let's discuss the unstained control. In order to unmix fluorochrome signatures from the multicolor sample, the background autofluorescence of the cells must be taken into account. It is important that the spectrum of the unstained sample matches the cells in the multicolor sample. Remember this when using certain tissues and when processing samples with different buffers and treatments. For example, when running an assay on mouse skin cells, spleen cells should not be used as the unstained control. Although their signatures appear similar, when we normalize the signal and compare them, we can recognize differences between the two. If we attempt to unmix multicolor skin cell data with the spleen unstained, there are unmixing errors present as extreme negative events. When we unmix with the proper unstained skin sample, these errors are resolved. Now let's review. Here is a control that meets all four requirements. 1. It has well-separated negative and positive populations and is at least as bright as the multicolor sample. 2. The negative population matches the particle type and autofluorescence profile of the positive population. 3. There are enough events to well represent the spectra of the negative and positive signals. And 4. The control and the multicolor sample have the same spectral signature. When each of these requirements are fulfilled for all of your reference controls, high-quality data can be achieved for any panel. Here, we can examine the NK cell populations identified in Cytek's 25-color immunoprofiling kit. Thanks to the use of quality reference controls, we can obtain clean orthogonal populations when identifying the HLA-DR negative population in the CD3 negative TCR gamma delta negative subset. Within this gate, we can clearly identify the early NK, terminal NK, and mature NK populations via CD16 and CD56 expression. Going even further, we obtain good resolution of the rare CD27 positive cells within the mature NK subset. This in-depth analysis within a 25-parameter assay is made possible by the use of accurate reference controls, following the guidelines discussed in this video. With that, we have concluded this lesson on the best practices for creating reference controls with the Cytek Aurora, Northern Lights, and Aurora CS. Whether you are using conventional or spectral flow cytometry, these are the best practices to ensure success and accurate data for your experiment. While this video covered the theory behind creating these reference controls, you can also view an in-depth tutorial on how to QC your reference controls in the SpectroFlow software. Visit Cytek's education page to learn more on this and many other topics.